Guys, I thought I would add a PowerPoint for you guys. This is typically what I go over in class because this concept can be really tricky, but I just thought I would go ahead and make one. So hopefully you guys are starting to get the hang of this online virtual learning, and hopefully you're starting to understand this concept. But if not, that's what I'm here for. I'm going to just kind of go through it, give you I know there's a lot of information out there from the different stuff we've looked at, and just kind of give you a breakdown of the more in a more simplified way. And then really that's it today. I did put an additional video that's really good to watch. And then you can just start working on your project. Okay, so the sarcomere. Okay, so I'm sure you guys have seen this picture. We've kind of already talked about the structure of the muscle. This is one single muscle cell called the myofibril. This is the actual cell, and then we pull it out to one myofibril that we were going to look at. And I'm sure you guys have seen this in pictures, but there's dark and light bands, and that has to do with the thin and thick filaments. The Z line, which you see right here, separates each sarcomere in a myofibril. And that is what you see shrinking when a muscle contracts. That is that Z line there, okay? So what makes up the sarcomere? We have the thick filaments, which are called myosin, and you typically on each side there's two thin filaments called the actin. And that's kind of what I want you. I know we have A bands, I bands, and you probably had that in your reading as well. Really, you just need to know the Z line and the thick and thin filaments known as myosin and actin. Same thing with the sarcomere. We need to know what that is as well. Okay, so structure, like I said, myofibrils are composed of two types of myofilaments. The thick one is myosin, where the thin is actin, and the arrangement of these filaments gives the skeletal muscle that striated appearance. So whenever we're making those on our mannequins, that dark band is going to be the thick myosin, where the light band is the thin. If we go back to our picture, you can kind of see the thicker lines versus the thin lines, the light lines, which would be the actin. Okay, myofibrils are surrounded by a special ER, and I'll show you a picture. This is called the sarcoplasmic reticulum. So they kind of have their special endoplasmic reticulum. And then we also have these things called T-tubules, and that runs through the muscle cell, and that's what basically allows for transport of necessary chemicals. Like when we get action potentials, that sends the signal to the actual muscle cell. So you can see here, this is our sarcoplasm. Each one of these is a myofibril. So if we kind of look at this, each one here is a myofibril. We have our actin and our myosin. And then the blue structure here, it's kind of like a webbing that goes over it. That is the endoplasmic or the sarcoplasmic reticulum. And then this yellow part here, these are the T-tubules. And so basically an action, which I'll show you in a picture later, an action potential comes down and it will go through the T-tubules, which we'll see will start the action potential in the muscle cell. And we'll kind of go through those steps in a little bit. But this is just kind of structure here. This would be like a fascicle with tons of myofibrils, and then this is pulling each one out here. Obviously, there's a lot of mitochondria. Muscle cells have a lot of mitochondria to provide a lot of ATP. Okay, myofibrils are divided into functional units called sarcomeres. They're bordered by the Z lines, and then, like we said, the thin filaments are actin. They're attached to the Z lines and they extend toward the center, where the thick filaments, myosin, are in the center. Okay, so this is one sarcomere. We have our Z lines here, which are kind of look like a little Z. You see the thick filaments. We have the myosin in the middle, and then the actin is attached to the Z lines. And what happens here is the actin on both sides kind of come closer to the middle, I guess you'd say in the H zone, when we see a contraction. Okay, so exactly what is the sliding filament theory? Basically, they don't change shape. That's kind of the biggest misconception. They don't change length at all. What they do is they slide past one another. So right here, it's relax, and then you see the actin on all levels come a little bit more towards the center when it is contracted, and we'll kind of go through how that occurs. Okay, so I'm just going to kind of read it first, which I know will be a little confusing, and then we'll look at some pictures to kind of go through it. So the myosin heads, they're bound to a site on the actin filaments, and they form cross bridges. ATP, the only way that we can actually have this work is by ATP. ATP is going to bind to the myosin heads, and it breaks that cross bridge. So when it is attached, ATP binds there and breaks that cross bridge. We hydrolyze it. Remember hydrolysis? Basically, ATP is used and it gets converted into energy, ADP, and phosphate. So once the myosin heads are attached to the actin filament, what happens is the ATP is going to be used and we get that energy. Now the myosin heads are released and they're full of energy, getting ready to perform again. So the energized myosin heads reattach to the actin filament, and then every time they do this, they're bringing the actin in, or the actin in a little bit closer, forming new cross bridges. And each one's doing this over and over super fast. Like I said, energized myosin heads release energy and push actin filaments towards the center of the sarcomere. This shortens the sarcomere, which is causing the contraction. To give you roughly an idea, each sarcomere has about 350 myosin heads. So each myosin head forms and reforms 5 cross bridges each second. So this is happening really fast. That's how we get the fast contraction. So I know that's kind of a lot. Let's take a look. Okay, so currently, here's our actin filaments up here, and then the thick one is the myosin. So they're currently attached up here. Once attached, ATP binds, should be bounded to the myosin head. Okay, so let's look down here. Here's our myosin head. It's low energy configuration as it's lowered currently. We look here, next step. So we just used ATP. So the ATP is going to join here. ATP is hydrolyzed, so we get some energy, and then it becomes ADP and phosphate, ADP plus B. Now it has high energy here, okay? Once it's also, see how it's kind of like in a cocked position? It's getting ready to basically throw up to the actin and help pull it. So now that it has the energy, it goes ahead and attaches to the actin and forms a cross bridge. And every time, and again, this is only one. Remember, there's 350. Every time a lot of them are doing it, it's going to pull it each time. Okay, now here, once a cross bridge is formed, it cannot be released until we get another ATP. Okay, we've already attached, we pulled it. It's now low energy again, and we're kind of waiting for an ATP so we can use the energy again. So ADP and the phosphate will stay next to the myosin head until and go basically to find another phosphate to become ATP. Then here, just low energy, waiting for ATP, we get another ATP, and we follow the same cycle. And each myosin head is doing that. Okay, so let's kind of recap that again. Essentially, we don't have any energy, we need ATP. Okay, once ATP's energy is used, we kind of cock back a little bit so we can attach, and it becomes ADP plus P, and we get energy. Now it's high energy. Once we have that energy, it is able to attach to the actin and form a cross bridge. And as it does that, the actin's moving closer towards the center. So up here, you would see these starting to move in. It cannot detach from the actin until another ATP comes. So ATP will bind, and then we'll keep doing the cycle over and over. Okay, muscles only contract when stimulated by a motor neuron. Okay, now let's talk about the next part here. It seems very easy with the actin myosin, but there's actually some things that get in the way, so that way we're not always contracting. We do need to have a relaxation period. So muscle contraction is regulated by proteins called tropomyosin and troponin, and or troponin, either way, you can pronounce it either way. At the resting state, the tropomyosin blocks myosin from binding to the actin. When it's contracting, the troponin opens the myosin binding sites. So these kind of help regulate so our body's not always in a contracted state. So let's say how this works. We talked about action potentials way back in Unit 2. Remember with the neurons passing We talked about sodium potassium pump. We're not going to go into the specifics about that, but that's kind of what's going on. So basically, an action potential travels down the motor neuron, and then the action potential is going to travel down those T tubules we talked about in the muscle fiber, so it can get to each myofibril. And then once the motor neuron triggers, so remember sodium channels open, and then it changes the voltage and the charge. We talked all about that. We're not going to go into detail with that. Hopefully, remember that. But as it gets to the T tubules in the sarcoplasmic reticulum, what happens is it releases a whole bunch of calcium. Okay, so here's our motor neuron up here. Does anybody remember? Hopefully, remember this is acetylcholine. Okay, acetylcholine gets released into channels, and that kind of stimulates. This whole yellow part is the muscle. That stimulates the muscle to send a signal, and it's going to go here, number 2. This is the synaptic cleft attaching. This is the neuromuscular junction. So it triggers a chemical reaction to the T tubules. So these are all going down the muscle. This blue part is the sarcoplasmic reticulum that stores a lot of calcium. So once this message gets here, it releases the calcium. Calcium then binds to troponin here. Okay, that's going to be the purple things. Once the troponin it attaches, it pulls up the tropomyosin, and then once with ATP, the myosin head can attach. Okay, now eventually we don't always want to be contracted state, but calcium will be recalled back for relaxation back to the sarcoplasmic reticulum, and then the calcium would no longer be bounded to troponin, and then tropomyosin will be blocking the site, so the myosin cannot attach, and that kind of helps regulate the reaction. So calcium binds to troponin. Tropomyosin exposes the myosin binding sites on the actin filaments, and the muscle contracts. When contraction is finished, calcium moves back to the sarcoplasmic reticulum, and binding sites are blocked. So that also helps regulate. So here's another better picture. Calcium comes in. This is when myosin heads cannot attach. So see that the troponin is in place. Tropomyosin is blocking where it would be. So the calcium will attach to the troponin, which pulls up the tropomyosin and now is exposed and ready for muscle contraction. Okay, so I know I'm going kind of fast, but I only have 15 minutes with this app that I'm using, so I have about 3 minutes left. The whole point of this unit is called rigor mortis, and that's actually what happens when you die. So a lot of people had questions about that, but in the process of death, there in about an average of 4 hours, no more ATP is being produced, which makes sense. Person is no longer living. So if there's no ATP, what's going to occur is that without the ATP, the myosin head cannot detach from the actin. Because remember, in order for the myosin head to detach, we need ATP. So if it cannot detach, it's going to leave the muscles in a state of contraction, constant contraction, and it makes them stiff. So that's typically why when you feel, well, hopefully none of us have felt a dead body, but if you would, the muscles are really stiff right after death because we can't detach. The myosin heads cannot detach. The calcium ions don't go back to the sarcoplasmic reticulum, so it just kind of is stuck in the contraction state until eventually your body starts to break down. But the average of 4 hours or more, there's going to be no more ATP, and then we're kind of in that contracted state. So hopefully that helped you guys a little bit. I know that was kind of fast. Make sure you know these steps here. Make sure you know the key players, troponin, tropomyosin, and calcium in this step. This is really good to know. Make sure you know all these, the T tubule, sarcoplasmic reticulum. And so guys, I did attach a video with Crash Course that has some really good visuals and does a really good job at going over this. So hopefully that helped. See you guys later.